assist them now that we have some tools that I feel like we can go and help them. So thank you very much.
Thank you, Brian. If I could, I would just suggest that we, if possible, let's hold questions and have them over lunch, to have conversations over lunch. Ray Beckham is going to talk about her work in microbial fuel cells.
Which one is that? Good afternoon. I'm going to speak about the work that I have been doing with Mike Ellis as a junior faculty collaboration research in the area of microbial fuel cells. Before I start, this was a completely new research area in my lab, so the support from ICTAS was extremely significant in establishing this area of research in my laboratory. Okay, so this research, as I mentioned, is about microbial fuel cells. A very brief introduction to microbial fuel cells. These are fuel cells that utilize exoelectrogenic microorganisms in order to oxidize organic or inorganic substance and generate power. They are a compact, portable, and sustainable source of energy, and they are, projected to be useful in applications that have to do with miniature vehicles, remote monitoring, and deep sea sensing. Now, the problem is that the amount of power these microbial fuel cells generate is too small to make them actually useful. So what we are doing here is to try to figure out some of the biotic and abiotic limitations that are there in these microbial fuel cells and try to address them. We are focusing specifically on the anode compartment of these fuel cells, and what we are looking to do is to improve the anode performance from two different directions. One is by improving the microbial attachment, and the other is by improving the electron transport from the bacteria that are attached. So for the rest of the 5 or 7 minutes, what I'm going to do is to first talk about the attachment and then about the electron transport. So from the attachment point of view, we have decided to focus on the initial step of bacteria attachment to the surface. The actual power is generated by a biofilm of bacteria, but for that film to form, you have to have bacteria adhere to the surface, strengthen that adhesion, and then proliferate and cover the surface. So what we hypothesize is that if we can understand that adhesion step and optimize that, then we should be able to optimize biofilm formation as well. In order to achieve that, what we did was to develop these model surfaces with well-defined and well-ordered surface topography and spacing between the topographies. And the take-home message is this plot right here, where, as you can see, by changing the spacing between the surface topography features and the size of the topography, we can control the total adhesion density of bacteria on the surface. So the key word here is to control. We can achieve minimum bacteria attachment density, which is not favorable when you're talking about microbial fuel cell. We want to, of course, have the maximum bacteria attachment so that we can have maximum amount of electrons transferred and maximum amount of power. So the point of design for microbial fuel cell is here, but nevertheless, we can control the adhesion for other applications that I will talk about at the end of the presentation. So we basically use this as our guideline for the design of electrode for the microbial fuel cell. Now, in collaboration with Mike and his AAP postdoc, John Wuhu, who is no longer at Virginia Tech, he just left in January, we conducted some electrochemistry experiments to see how these surface topographies are affecting electron transport. Our focus, as I said before, was to improve the anode. So instead of working with a whole fuel cell, we worked with a 3-electrode system, as you see here. The working electrode that we are using is glassy carbon, and we basically generate roughness on glassy carbon of different orders, a 10 nanometer electrode and another 100 nanometer roughness electrode, and see what is the electrochemical performance of these electrodes. And as you see here, for example, the current density, there is a 4-fold increase in current density when we go to 100 nanometer roughness surfaces, whereas the difference in the total electrochemically active available area increase is only 20%. So it's not that we have increased the area. Yeah, of course you will increase your current. It's beyond that. It's beyond the simple increase in area. And this is further supported by looking at electrochemical impedance spectroscopy. And as you see, the charge transfer resistance for the smooth electrode transfer resistance for the rough electrode. And similarly, the surface coverage by bacteria is a function of the roughness of the surface. So basically, by controlling the surface roughness, we can control the two key components that affect microbial fuel cell performance, which is the adhesion as well as the electron transfer. And we believe that we have demonstrated this in a very methodical manner for what we believe is the first time. The adhesion component, we have published it in Soft Matter towards the end of last year, and the electrochemical performance that I presented, we presented this at the ASME annual conference last November, and we are working on a journal submission right now. Now, all that I talked about was focused on improving the electrode surface, but we believe that the whole story focuses on interaction between the bacteria and the electrode. So looking at the bacteria and trying to optimize that is equally important. So we have started collaborating with Dr. Vishwarup at BBI to basically look at how these changes in surface topographies are affecting bacteria, gene expression, and physiological properties. So we have actually written a proposal to NSF, submitted this past cycle, to look at this unified approach of both surface engineering and genetic engineering. So using, building upon what you have seen here and combining it with genomics and proteomics approaches to hopefully optimize both the bacteria and the surface to truly optimize the anode compartment of the fuel cell. We have also submitted another proposal to Air Force in the past, which unfortunately was not successful. We continue to speak with them to see how we can improve our proposal, and with the help of John Green, we have been in touch with the SPAWAR research labs to see, in collaboration with them, they seem to be open to that idea of writing a proposal to ONR. Now, when I started talking about all this, I mentioned the controlling of adhesion. So if we can control the adhesion, we can increase the adhesion for microbial fuel cell application, but also for other applications such as, say, bioremediation. We can also reduce or control the bacterial attachment in order to have minimal bacterial attachment to the surface, and that is relevant if you're looking at infection of biomedical implants, host-pathogen interactions, and that kind of stuff. So that's all the other areas that are related, and in fact, if we quickly go to the next slide and come back. So this is, based on the work that we did for this project, this is one of the spin-off directions where we have a smooth polystyrene. Surface polystyrene is one of the commonly used materials for disposable biomedical devices, such as catheters and things like that. If we roughen the surface with these nanofiber structures of the right diameter and separation distance, as you can just qualitatively, visually looking at it, you see that you have significantly smaller number of organisms on the surface that is textured with nanofibers. Okay? So that's another direction that we are pursuing. And of course, as everyone else mentioned, students play a big role in these works. And the students who have been working on this project have been recognized both for their research work with People Award from Adhesion Society and also an undergraduate research award from the Adhesive Manufacturers Association, and also academically with Cord Fellowship from the Mechanical Engineering Department. And lastly, we have also filed an invention disclosure for the adhesion part of this research. Thank you.